Hey, I'm Dr. Rob. Welcome to Biblical Genetics. You know, in my unending quest to find an interesting place to film, I ended up at the Great Sand Dunes National Park in Colorado. I'm here for a week to speak at different churches and creation groups, and I had a day off. I was planning on doing some snow activity, but I landed in Denver. It was about 70 degrees and nary a flake to be seen. Today it's about 30 degrees. The wind just picked up as soon as I pulled up, so it's quite cold. I'm going to try to do this a little quickly because it's chilly. I want to talk about a new paper that just came out, a paper that actually duplicated something that I tried to do nearly 20 years ago with a limited data set. It's Lake et al. 2024, Quantifying Constraint in the Human Mitochondrial Genome. It's a very interesting question. How much of the mitochondrial genome can change? So we're talking about Mitochondrial Eve. We're talking about the history of humanity, possibly a human-chimpanzee common ancestor, and what would have changed in that time since that common ancestor for the evolutionists? And what should the creationists think about the Mitochondrial Eve question? This is a very interesting paper because it actually duplicates something that I tried to do nearly 20 years ago with a very limited data set. They have a huge data set now. They're able to do things that I could never even have imagined. But this is informing us in some ways about the creation evolution debate, because how much of the mitochondrial genome can mutate? And when we see mutations, what direction do they go? Are they predictable? Can we forwardcast the changes that will happen in a genome? And if you reverse that by billions of years, what should the genome should have looked like? Well, you know what? The answers actually aren't very evolutionary, but we'll get to that. This Lake et al. 2024 paper is following up another paper by Laricchia et al. From the same laboratory. This is 2022. Mitochondrial variation across 56,434 individuals in a thing called NOM-AD. 56,000 mitochondria. That's amazing. They actually started with 90,000 and had to pare it down because a lot of the data were bad, which is something that I saw way back in 2007. My paper was Mitochondrial Diversity Within Modern Human Populations. I'm very proud of this. I worked really hard on this. I was at the time working with John Sanford. He was overseeing this project, but I did all the data analysis and I wrote most of the paper, so my name is on the paper. I actually got 17 citations. Oh, wow. Now, I know that doesn't sound like a lot, but most scientific papers get no citations, or they get one or two, and it's usually a citation by the same person who wrote that paper. They read another paper and they cite themselves, or the same laboratory might cite it. So most papers don't get a lot of citations. I did get 17, but not only that, I get 12,000 downloads, or at least 12,000 people who read my abstract, and many of them would have downloaded the paper. So I know that the movers and the shakers in the mitochondrial genetics world read my paper. Back then, we didn't have a lot of data. I had just under 2,000 mitochondrial sequences, but I had to do a lot of data cleaning up also. I had to reject most of them. I ended up with 827. In fact, all of the sequences that were being used by the US FBI were garbage. And I pointed out, these sequences are full of errors. You can't use them. So I'm assuming they've cleaned them up since then. But in those early days of DNA sequencing, a lot of mistakes were happening. And they were propagating into forensic databases. That's not cool. But what I was trying to do was several things. One, I was trying to calculate Mitochondrial Eve. Now, Mitochondrial Eve was discovered in 1988. They had done not full genome sequencing, but just looking little bits and pieces of the mitochondrial genome from people around the world. They said, wow, look at that. There was a single human ancestress of all people alive today. Very interesting. So I was trying to calculate the Eve sequence because no one had ever done it before. But I only had these 800-something sequences, and most of them were from Europe. And I knew that. I knew it was a problem. I knew I could not actually get to Eve necessarily because I had a sampling problem. I had mostly European sequences. So what I did was I took what's called a consensus approach. I just took the average. I lined up all these mitochondria, which took a long time, because it's 16,569 letters long, and I had to line them up. And then I took, for each column, I just took the average. So if everyone had an A, there was an A there. If 99% of the people had a G, there was a G there. And that instantly gets rid of all the rare variation. The problem with that approach is that it depends upon your sampling, in that my Eve calculation was a European-like sequence. Of course, mostly European sequences. If I take the average, it's going to be European. Like today, if you looked at all 8 billion mitochondrial genomes in the entire world and you took the average, I'm pretty certain it would be someone walking down the streets of New Delhi today. They'd be the average. Why? Because there's more Indians than anyone else in the world. That's why it's a sampling issue. A consensus is not the ancestor, but it's the best we could do at the time. And I said in the paper that as we add more sequences, specifically sequences from Africa, Eve will change. Not a lot, but it'll change toward a more African-like sequence because we didn't have many at that time. But I did show that 84% of the sequence was invariant. In other words, for that letter in the genome, 84% of the time, every single person in my sampling database had exactly the same letter. Oh, that's very interesting. That means that a lot of the genome can't actually mutate, which informs us about evolution. If it can't mutate, then evolution is stuck. And I also showed that some mutations were much more common than others, which also informs us about evolution. Because if some mutations are more common, that means that mutations go in a certain direction, specifically the C to T mutation. Cytosine, C, very often will lose an amine group, this NH2, amine. And when that happens, it now looks like a uracil. And because of the chemistry and the DNA repair enzymes that happen, very often the cell will replace that then with a T. So the C to T mutation is the most common mutation. So one wonders that if mitochondria have been around for 1 billion years or so, whatever the evolutionists say, and C to T is most common, how come all the C's haven't mutated into T's in 1 billion years? Because the reverse mutations are not happening. Oh. So it looks like the mitochondrial genome is not really as old as they think it is, and it looks like it's predictable, which it's not supposed to be, because evolution is not supposed to be predictable. All the things are supposed to be on the table. But no, when you actually look at the mutations, they're all going to go in a certain direction. So that was my paper. Interestingly, I got a very negative citation, which I'm going to talk about. This tells us a little bit about the mindset of the evolutionist. Well, let me go through some history. In 1981, Anderson et al. Published what was called the Cambridge Reference Sequence. Now, it wasn't called that. It became called the Cambridge Reference Sequence. It was the first fully sequenced human mitochondrial genome. It was a milestone of scientific advancement. 1981, though, I think their work was probably manual. We didn't have the modern sequencing machines that we have today. And they came out with the sequences, was 16,569 letters long. But it had some errors. In 1999, Andrews et al. Redid it. They resequenced that same sample, and they found that some of the letters were wrong, and they found an extra letter. And that was a big problem because all these other papers had now been published in mitochondria, and they're saying, okay, now at letter number 9002, there's an A, and that's in such and such a gene in Africans versus Europeans. And, well, if you take out that letter in the reference, then all the letters downstream are going to change, and all the numbering gets messed up. And it was in like 300, in the 300s range. So literally, most of the genome would have to change. They didn't want to do that. So you know what they did? They put an X in the sequence. Yeah. And that X, all these years later, is still there. In fact, I just saw it in the Laricia et al. 2022 paper. They have a chart that's showing the coverage in other words, how many copies of or how many times the mitochondria was sequenced in the database that they're looking at per individual. And it goes along, and right in the 300s, it drops down to 0 and pops back up again. Yeah, that's the X. It's still there. Oh, that's crazy. Okay, 1981, Anderson et al., the Cambridge Reference Sequence. 1999, Andrews et al., the revised Cambridge Reference Sequence. 2007, I tried to publish Mitochondrial Eve, knowing that it would have to change over time. But then, a few years later, a very important group of mitochondrial scientists, Behar et al., 2012, they wrote a paper called A Copernican Reassessment of Human Mitochondrial Tree from Its Root. And they gave us what was called I think it's the Revised Sapiens Reference Sequence, sapiens being Homo sapiens. What they suggested was, we got to get rid of this Cambridge Reference Sequence entirely. Let's use an evolutionary route. I'm wondering if they saw my Eve paper and realized that was a European thing. Oh, we can't have that. Let's use the out of Africa. That's going to be our reference sequence from now on. And well, it didn't go very well because everyone's still using the RCRS, but they had the RSRS, Revised Sapiens Reference Sequence. Now, 2 years later, Bandelt et al. Now, Bandelt is a big mover and shaker in the world of mitochondrial genetics. He's written a lot of papers. I've referenced a lot of his papers. 2014, the case for the continuous use of the Revised Cambridge Reference Sequence. Now, in here, he cited my 2007 paper unfavorably. Now, I'm a big boy. I can handle I can handle criticism, and I can also handle the fact that he completely misrepresented what I said, or maybe even never even read the paper, but whatever. In the middle of this paper, he's talking about reference sequences versus consensus sequences, which is an average, versus a phylogenetic sequence, which would be the RSRS. And they wrote, as a curiosity, not only a reference sequence, but also the root of the mitochondrial phylogeny has been confused with a consensus sequence. No, it hasn't, but that's what they said. Derived from some sample set deemed to be representative of worldwide mitochondrial DNA variation. Well, actually, no, it wasn't deemed. It was all the variation that we had. Literally, we had every single mitochondria available in the world at that time in our database. And it was from various people around the world, knowing fully well that was weighted toward the Europeans. Okay, but they keep writing. This was cited in an article, and they cite my 2007 article, which was actually following the agenda of creationism. What agenda of creationism? But here they cite my International Conference of Creationism paper, or Dan Criswell and John Sanford were my co-authors, where we take that idea of mitochondrial Eve and talk about it in a creationist perspective. What agenda of creationism? My 2007 paper was simply an analysis of the raw data. Are you afraid of the raw data, Mr. Evolutionist? No, I don't think so. I think they're just trying to throw me under the bus, but that's fine. They continue. Using a strongly Eurasian-based sample, which we admit in the paper, the root of haplogroup R was then offered as the root of the global mitochondrial DNA phylogeny. Now, this is kind of true and kind of not true at the same time. Specifically, in my ICC paper, at that time, I was thinking that there's a place in the phylogenetic tree where you can see 3 branches. That would be the 3 daughters-in-law of Noah. And that was right around this R area. Now, R is just a name of a branch. There's A, B, C, D, E, G, R, S, T. They named all the different branches on the tree. And R happens to be associated probably with like the Yamnaya people who gave us the Indo-European languages. Indo-European languages. And when they split East and West, that's why Hindi has similarities to like French, because of this common ancestral population from Central Asia, which gave rise to major world populations. That's probably what R is, or somewhere in that region. I didn't say that that was the answer, but I was thinking that there should be 3 branches on the tree, except I don't believe that anymore, because I realized that if Noah's daughters-in-law were all sisters, there'd be only 1 branch on the modern mitochondrial family tree, or 2 of them were sisters and 1 wasn't. We might get 2 branches on the modern mitochondrial family tree. Plus, we don't know anything about the mutation rates before the flood. We don't know how much diversity there should be. You actually can't know from first principles, looking at scripture, if there should be 1, 2, or 3 branches on the tree. So I've changed my mind there, and that was kind of what I was thinking in that paper. But they flagged that, and that's fine. I can change my mind, but there was nothing wrong with the paper itself, either paper. I'm just trying to figure out what the Eve sequence is, how close we can get to it, and then how constrained is the genome. And that's what these 2 new papers, the Lake et al. 2024 and the Larici et al. 2022, they looked at how much variation we see. Now, there are 56,000 human genomes. They showed that about 50% of the human mitochondria, it never changes. They never saw any variation at that spot. Whoa, 50%. So half of it probably can't mutate, or the individual dies. That's why you don't see it, because the mutations are happening, just you never see them. They also showed that of all the letter changes in the genome that would affect the protein, the amino acid in the protein, that you can have what's called a synonymous change or a non-synonymous change. A synonymous change is when you change the letter in the DNA, but it doesn't change the amino acid in the protein. Because as you know, when proteins are manufactured, they're manufactured from a messenger RNA. And there are codons in the messenger RNA. 3 letters in the messenger RNA is a codon, and each codon specifies which amino acid goes in the protein. But there are 64 possible combinations of 3 letters. If you have 4 times 4 times 4. But there's only 20 or 21 amino acids. So most amino acids have more than one codon. So if you can mutate the codon, very often it doesn't change the amino acid. But out of all the possible synonymous changes, they only saw about half of them. They only saw about 10% of all the possible changes, mutations that would change an amino acid, which means most amino acid changes are bad. Yeah, it can happen, but they only saw 10% of them. And they only saw about 1% of all the possible mutations that would stop a protein earlier, or if you change the stop codon, make the protein go longer. So there's constraints there. But then in the Lake et al. Paper, it's amazing stuff. They showed that specific areas of proteins do not change, and they figured out why. They showed that specific areas of the RNAs don't change, and they showed why. In my not the splicing and dicing one. Oh, in the episode that I did called the spaghettification of irreducible complexity, I talked about in the middle of that episode how the transfer RNAs that have on the bottom an anticodon, the 3 letters that matches up with the codon in the on the RNA, the anticodon, and then the top they have an amino acid, and they're used in the ribosome to manufacture the proteins. But those transfer RNAs are heavily modified. They're modified sometimes 15, 20 times. So another protein will come and attach to it and change the letters of the RNA or stick something on it. And what Lake et al. Showed was that in the anticodon, you can't change the first 2 letters. The 3rd letter, that's more changeable, because when you have an amino acid that's 4 different codons, it's the 4th one, the wobble base, that can be different. It can be A, C, G, or a T. So if you mutate that one, you keep the same amino acid. But if you change the other ones, you change the amino acid, that's probably bad for the organism. They showed that in the places that get changed on those transfer RNAs, those places are pretty much invariant. Oh, so the cell needs that letter there, so another protein can come and attach to it and make a chemical change to the RNA. Or the RNA doesn't function right, and the protein doesn't get made right. So there's a lot of constraint. There's a lot of evolutionary, if you want to call it that, there's a lot of evolutionary design systems, whatever you want to say, constraint. You can't just change anything. Only certain mutations are allowed. Oh, but they also showed that specifically the C to T mutation is by far the most common mutation. In fact, the C to T mutation on the coding strand of the mitochondrial genome, not the opposite, the anti-coding strand, whatever they call it, the strand that doesn't actually code for the RNA and the proteins. No, it's the C to T happens on the coding strand way more often than anything else. So, given billions of years, how come all those C's haven't changed to T's? That's a great question. So, what do we know here? 1, the mitochondria is highly functional. 2, mutations are predictable, which are not supposed to be. 3, it doesn't look like the mitochondrial genome is billions of years old. 4, mitochondrial Eve is a very difficult thing to figure out because it depends upon where in the tree you want to place her. Now, I think she's at the center of the so-called starburst, which is the radiation of all the branches from some central point. I don't think she's on an African branch, specifically also because we've already shown, and I've said on this channel before a couple times, that mutations are different along different branches, meaning that you can't use a molecular clock to predict where Eve would be. Okay, so that's it for this episode. I am freezing cold. I get back in the car. I got about a 2-hour drive back to Colorado Springs. Thank you for people on podcasts listening. Thank you for people on YouTube and the other video channels watching. Thank you for my supporters. I really appreciate you guys. If you'd like to help support the show, if there's some links in the notes, you can easily find that. But just have a blessed day and continue to explore God's creation, because there are amazing things still waiting to be discovered.